German botanist Schleiden proposed that all plants are made up of cells. And after Schleiden, Theodor Schwann stated that all animals are made up of cells. What did he observe? He observed nuclei in all types of animal cells, but he failed to observe cell wall in them because cell wall is the part of plant cells. It is not present in animal cells. All right. Then Rudolf Virchow in 1858 stated that new cells come only from other cells. Animal cells come from animal cell and plant cells from plant cell. All right. So look at the names of the last three biologists, Schleiden, Theodor Schwann and Rudolf Virchow. Their combined statements is known as cell theory. Alright, so what are the postulates or salient features of cell theory? All living organisms are composed of one or more cells. The cell is the smallest, basic, structural and functional unit of all organisms. It means that an organism cannot work without the function of the cell. Okay, every function happened at cellular level. Okay, respiration, digestion, everything. Third point, new cells are formed by the division of pre-existing cells. Alright, so that was the postulates or salient features of cell theory. Now because we observe the cells with the help of a microscope. There are two kinds of microscopes, light microscopes and electron microscope. You can see the pictures below, and in your laboratory you can use light microscope easily. The name is showing that light microscope uses light and electron microscope uses a beam of electron. Okay, so light microscope uses visible light as a source of illumination. Electron microscope uses beam of electrons as a source of illumination. In the light microscope there are glass lenses for magnification, but in electron microscope it uses electromagnet in place of lenses. The image can be focused in human eye when we see in light microscope, but in electron microscope the image can be focused in human eye. Therefore photographic plate or screen is used. Alright. Light microscope can magnify the object up to 1000 times than the real size of the object. But electron microscope's magnification power is 10,000 times greater than light microscope. Alright. Light microscope resolving power is very limited, just 0.2 micrometer. Resolving power of electron microscope is 0.2 nanometer and it reveals organelles. We cannot see the cell organelles that are present parts of the cells with light microscope, but when we can easily see with electron microscope. So what are the pictures we are seeing in our book or internet? They all are taken from the electron microscope. So this is the comparison between light microscope and an electron microscope. Now there are two words, magnification and resolution. We must clear the meanings. So what is the difference between resolution and magnification? You can see these two red pictures, capacity to separate adjacent objects. Alright, this is called resolution. Or you can say the clearance of the, how much is the image is clear. What is magnification? It is basically the increasing size of the object. Okay, a small size will look bigger. Second point of resolution, it is maintained up to certain magnification. And with magnification, resolution is disturbed. When we magnify the object, size of the object will be increased, so this will be not so much clear. Alright, how can we improve resolution? It improves as wavelength of illumination becomes shorter. Okay, the light we are using, its wavelength should be shorter, so resolution will be more. And magnification improves with the focal length of the lens. How much focal length is of the lens is there, greater will be the focal length, the magnification will be the high. Alright, so this will This is the comparison between resolution and magnification. We must